Hello, champions, and welcome back to PW English. And I'm your Diksha ma'am. So, here we are learning about the neural system of our body, and we have started our chapter Neural Control and Coordination. So, let's talk about the other part of Neural Control and Coordination chapter. So, what we are going to cover today, and what we have actually covered in the previous lecture. In the previous lecture, we have learned how the neuron functions. We have talked about how a nerve impulse is generated, conducted, and transmitted. Also, we have discussed about the nervous system, various parts, and brain, which is the most important part of your nervous system. So, today we are going to start with your spinal cord. Let's talk about spinal cord. So, if I talk about spinal cord, where it is present, first of all, it is present in neural canal of vertebral column So in our vertebral column, that is in our backbone, we have an empty space, and that empty space contains the spinal cord, right? So the spinal cord has the length shorter than the vertebral column, because what happened when we were growing, our spinal cord, it stops its growth at the age of around 5 to 6, whereas vertebral column keeps on growing because your height increases. So we always say that the length of spinal cord is always shorter than the vertebral column. So if you will see the spinal cord clearly, you will see the swellings on the spinal cord. One is a cervical swelling and another is a lumbar swelling. Yes. So the portion of the spinal cord has been, demarcated depending upon the vertebra it is present. So it ends around L1 vertebra. This is if L1 and this is L2. It will end around L1 and L2 vertebra. Okay. So the end part of spinal cord is known as conus terminalis. Conus terminalis. So if someone asks you what is the end part of spinal cord, you will say conus terminalis. Okay. So this is the swelling, which is a cervical swelling, because it is the portion of spinal cord that is present near the cervical vertebra. So this is cervical swelling, and this is the lumbar swelling. Okay. Now here, as your brain was covered by the protective coverings known as meninges, the spinal cord is also covered by the protective coverings known as meninges. So likewise, our brain had the meninges, dura mater, arachnoid mater, and pia mater. It also has the meninges, pia mater, arachnoid mater, and dura mater with the same sequence. Okay. So the innermost covering, that is pia mater, it extend down like this, and this portion of spinal cord is known as filum terminale. So if someone asks you what is the end part of spinal cord, you will say conus terminalis, not filum terminale. Why? Because filum terminale is just the extension of pia mater. So, as you know, the nerves emerges from brain as well as spinal cord. So, at this portion, the nerves, they emerges like this, and this appears like the tail of the animal horse. So, this portion you also call it as cauda equina. Now, you must be confused, ma'am. Wait, this is too much. Wait. So simple. The end portion of the spinal cord is conus terminalis. The extension of pia mater at the end of spinal cord is filum terminale, whereas the arrangement of nerves that are emerging from the last portion of spinal cord is your cauda equina. So this is arrangement of nerves. Okay, I hope that's pretty clear to everyone. Now let's dissect the spinal cord. Let's dissect it. So when we dissect it, so you will see such kind of a thing. Okay. If this is a dorsal side, which is also known as posterior side, this one side. Okay. And this is the ventral side, this one also known as the anterior side. Alright. And in the center of your spinal cord is present an empty space which contains CSF. What is CSF? Cerebrospinal fluid, right? So this portion is known as central canal. Central canal. It contains CSF. I know you all know what is the full form of CSF. Cerebrospinal fluid. That was also present in the brain and it was formed by the filtration of blood. Okay. Now, if you will see the portion of the spinal cord, it will also be having the gray and the white matter. Like in brain, the outermost layer was of the gray matter and inner was of the white matter. Here in the spinal cord, it's opposite. In the spinal cord, the center portion is your gray matter. The center portion is your gray matter, whereas the outer portion is your white matter. All right. So here, remember, the outer is white matter and inner is gray, just opposite to that of a brain. So you can see this portion is your gray matter and this one is your white matter. Okay. So the areas of gray matter, they are known as horn. What do you call them as horn? So this portion is your posterior horn. And this will be your posterior, the white matter, sorry, anterior, the white matter will be known as funiculus. Okay, so let's make it simple. The area of the gray matter is known as horn, whereas area of white matter will be known as funiculus. Just like on the anterior side, we have area of white matter, which is anterior funiculus, and this is anterior horn. So from these matter emerges your nerves. What emerges? Nerve from both the side. From both the side, the nerves will emerge. Okay. So from this dorsal side, sensory nerves will emerge. Which nerves will emerge? Sensory. And from the anterior side, motor nerves will emerge. Which nerves will emerge? Motor nerves will emerge. Okay. So in the sensory portion, when the sensory nerves leave the spinal cord, here they will meet into this portion which is known as ganglion. It is known as ganglion. So we have done this in the structural organization in animals, that this dorsal root ganglion of spinal cord contains pseudo-unipolar neurons. So this is this ganglion which contains pseudo-unipolar neurons. Now these both nerves are going to meet here. These both are going to meet here. And what are they going to form? They are going to form spinal nerve. They are collectively going to form the spinal nerve. Okay. So can I say the spinal nerve contains both neurons of sensory and motor neuron? Yes. Yes. Can I say the spinal nerve which is forming here contains both sensory and motor neuron? Absolutely correct. Because it is formed by the combination of the spinal nerve, sorry, sensory nerve, slip of tongue, pardon me, sensory nerve and motor nerve. So we say that our spinal nerves, they are mixed in nature. They are mixed in nature and there are around 31 pairs, 31 pairs of spinal nerves in our body. So how we say it in pairs? Because as I've told you, the nerves are emerging from both the side, from here as well as here. So total spinal nerves are 31 pair and the nature is mixed. That means it will be containing both sensory as well as motor neuron. Okay. So that's the section of your spinal cord. Now, the most common question that is asked in the NEET exam is about the portion. They will ask you which of the following portion will contain the sensory portion and the motor neuron or the motor portion. So dorsal posterior is always sensory. It is always sensory. Okay. Whereas ventral anterior is always motor. Okay. Once the question asked was, what if the posterior horn gets destroyed? Which of the following impulse will not emerge? Sensory or motor? Because if posterior is getting destroyed, the sensory neurons will not send the signal. Okay. Got it? So remember DPS, Delhi Public School are very common, these schools in North India. I know a lot of students are from Nepal or or from Northeast or from South India, but I tell you there is a school named Delhi Public School. So from there, remember DPS, Delhi Public School, dorsal, posterior, and cell. Okay, I hope now you will never forget this, right? Yes, boys and girls, this is going to be your last class of champions. So be enthusiastic. Yes, okay. So, like we have spinal nerves, we also have cranial nerves. We also have what? We also have cranial. So cranial nerves are the nerves that emerge brain, from brain. Like spinal nerves were 31 pairs, these are 12 pairs. Spinal nerves were mixed in nature. These can be sensory. These can be, some of them can be motor, and some of them can be. Okay, you don't need to remember all the nerves because they do not ask question about all the types of nerves. So there is no need to waste your time, right? Okay, now let's talk about reflex action. First of all, what is a reflex action? In your lower classes, you must have heard of that hot plate thing, in which when a person touches a hot plate, the arm itself gets back. This is a kind of reflex action. So technically, reflex action is your unplanned, that means you have not planned it, you have not thought about it. Sudden, that means it will not take much time, it's just spontaneous. Sudden, unplanned, sudden response to a stimulus. Okay, it's unplanned, sudden response to a stimulus. You have not planned it, you have not thought of it. For example, when you touch something, you are not aware that thing is hot, but suddenly your hand moves out. Have you heard of it that something will be hot and I will put my hand away? No, your body itself is functioning here. So this type of actions in your body, they are known as reflex action. Okay, so these reflex actions in your body, they can be conditioned as well as unconditioned. They can be conditioned as well as unconditioned. What's the difference? So conditioned reflex action is a type of reflex action that you learn during oh my god, slip of tongue is so much today, yaar. You learn throughout your life. For example, you are listening to me, and some of you must be listening to me, learning as well as writing. So you are doing all these things simultaneously, right? This is the kind of reflex action that you learn in your life. Or you can say that those who are in navy or army, they are learnt of three shots, one, two, three. So that becomes their reflex. Okay, so whenever they're going to shoot, they will shoot and they will shoot like three times. That's a kind of reflex. Okay, so some reflexes you learn throughout life, like the mothers, what they used to do, they're weaving, they're knitting things like this, and simultaneously they are also watching the TV and talking to you. So this is also a kind of reflex. Okay, so conditioned reflex are reflexes. These are reflexes that are learned during life. Learned during life. Okay, whereas unconditioned are not learned during life, they are since birth. For example, uh, if we uh, if we touch something, we have not learned it, the body will respond itself and it will take away your arm. If you touch anything hot, and your body will respond to it and it will take away your arm. Or when a if I show torch, or put the harsh light on the eyes of a newborn baby, and the eye will get closed, that's also a kind of reflex. So that child has not learned it, it's just since birth, it's in the body, the body is reflex. Okay, so that type of reflex actions, they're known as unconditioned. So now here in reflex action, what are the body organs involved? So first of all, the thing that is involved is sensory receptor. Sensory receptor. What are sensory receptors? These are certain cell-like structure which are attached to the neurons. Which are attached to the neurons. And these neurons, for example, if this is receptor, these neurons send signal to your central nervous system. Which nervous system? Central nervous system. So hence central nervous system is also involved. Like on your skin, there was a sensory receptor. When you touch anything hot, the sensory receptor will is attached to sensory neuron, and it will send signal to my central nervous system. It can be brain, it can be spinal cord. So out of this, the reflexes, there are different reflexes have different types of area in central nervous system. Like the one I'm talking about, hot plate one, that is present in spinal cord. Whereas visual reflexes, they're present in your brain, in your midbrain, right? So here, brain can be a part of it, even the spinal cord can be a part of it. Now, so from sensory receptor, which neuron is giving impulse to the CNS? This is afferent neuron. What are afferent neuron? These are sensory neurons. Okay, so most of students were telling me in the comments in the previous lecture that ma'am, you have not covered afferent or sensory neuron. Please go through the lecture very carefully. I have covered there about sensory, efferent nerve fibers. Please watch the entire lecture, right guys? Watch the entire lecture, then write some things. So you, if you are writing these things which are already present in the lecture, that's really not good. That means my hard work and my efforts are going in vain. So please first watch the lecture carefully, then write something. Okay? Okay. Okay. So here, the impulse from the central nervous system, now it is going to your effector organ. All right. Now, as I've told you, the you have touched the hot plate. Now the sensory neuron will send information to CNS. CNS through motor neuron will send information to your arm muscles, and muscle will take your arm away or your hand away. Okay. So here the information with the help of motor neuron or efferent neuron or nerve fibers will be sent to your organs, and these organs can be anything. These organs can be your muscles, both the muscles, smooth and skeletal, as well as your organs, visceral organs. So this structure that is formed here involving different organ, you call it as reflex arc. What you call it as? Reflex arc. So this entire structure that is formed, you call it as a reflex arc. Okay. So in the reflex arc, what do we have? The sensory receptor attached to sensory neuron giving information to CNS, and from CNS through motor neuron, the information is sent to the muscle or the organ. The very good example of this reflex action given in your NCRT is your, yes, knee jerk reflex. So let's talk about the knee jerk reflex. Knee jerk reflex. What is this knee jerk reflex? For example, if this is your knee, this is your leg, this is your thigh, this is your leg, and this is your knee. And in knee we have a bone known as patella, like this. Okay. So what, first of all, let's see what are the various muscles present in your thighs. So here we have muscles known as quadriceps muscle. And here we have muscles known as hamstring muscle. Okay. This muscle is quadriceps. And this muscle is hamstring. The function of hamstring muscle is to keep this leg in a bending position. So whenever I am bending my leg like this, if you can see here, the leg is at the bending position, the hamstring is functioning, or it is contracting. If I want to straighten the leg like this, then quadriceps will work. What will work? Quadriceps. So these both are what? These are muscle. Quadriceps muscle to straighten the leg, and hamstring muscles to bend the leg. Now sometimes what happen is, accidentally if you hit your this particular portion of the knee, or there is a doctor hammer, if doctor wants to demonstrate reflex action, the doctor will hit your knee with the hammer. Now that hammer is a doctor's hammer, not your home hammer. I maybe your some students who are very naughty, they will put the hammer and started doing the experiment on their kids. Please don't do it. Not the kids, not their kids, I'm sorry, on your younger brother and sisters. So please don't do this at home. This is really risky. The hammer that doctors use is different from the one we use in our home. Okay. So here what happen is, if accidentally you get hit here, what happened at that point is, you have tendons present here. What is a tendon? That connect muscle to a bone. And these tendons, they have sensory receptors. These tendons have sensory receptor. Now these sensory receptors have sensory neuron which will send information to your central nervous system. As you can see here, it will send information to your central nervous system. In the central nervous system, your sensory neuron will form a synapse. It will form a synapse with the motor neuron, and this motor neuron will go to quadricep muscle and ask the quadricep muscle to contract so that this leg should move in this direction. Got it? What will happen if your tendon gets stretched because you get hit or accidentally it gets stretched? The sensory neuron, which neuron? This neuron is sensory neuron or sensory nerve. It will send information to the central nervous system here, wherein a synapse is formed. A synapse is formed between sensory and motor. Now motor will give information to the quadricep muscle, and this muscle will contract and they will be straight leg. Okay, this is the reflex action, which is a knee jerk reflex. One reflex arc involves one reflex action. So this is your knee jerk reflex. So you can see in knee jerk reflex in the central nervous system only one synapse is formed. You can see here only one synapse is formed. So we say that the knee jerk reflex is a mono means single, synaptic, one synapse reflex. It's that reflex in which only one synapse is formed. Okay, this is one reflex action known as knee jerk reflex. Now, meanwhile here, another reflex action is also functioning or working here, which involves another neuron in the central nervous system known as interneuron. So now sensory nerve will form synapse with interneuron, and now this interneuron will form the synapse with motor neuron, and motor neuron will go to hamstring muscle, and hamstring muscle will be inhibited. Why it will be inhibited? Because we want to straighten the leg. If you want to straighten the leg, that means this helps in bending, so this should be inhibited. So here the inhibition of hamstring take place. This is another reflex, since it involves another neuron known as interneuron. So you can see there are two synapses formed. So this type of a reflex is a polysynaptic reflex. Polysynaptic reflex, because two reflexes are present there, or they are formed there. Okay, I hope that's pretty clear to everyone. So this is your reflex action, boys and girls. So do not get confused about polysynaptic and monosynaptic now. All right. Okay. So this was about your reflex action. Let's talk about autonomic nervous system, ANS. ANS stands for autonomic nervous system. Why you call it as autonomic? Because it is automatic. It's not under our control. The autonomic nervous system is also known as visceral nervous system because it innervates visceral organs. Okay. So now this ANS or autonomic nervous system, the CNS or autonomic nervous system has two parts. One is parasympathetic, and another is sympathetic. Okay. Now, what this autonomic nervous system is made up of. So we'll start with the CNS here. This is your central nervous system, and from central nervous system emerges the cranial and spinal nerve. So technically, ANS or autonomic nervous system is a part of peripheral nervous system, and peripheral nervous system involves all the nerves that arise from central nervous system. Okay, so now what happens is, from central nervous system the nerves are emerging, and these nerves definitely will be carrying the neurons. So these neurons now here, because they're not that long enough, so they have to go somewhere and give a halt. For example, if this is your visceral organ here, okay, this is your central nervous system. So from central nervous system, the nerves are emerging having the neurons here. So since they are shorter in length, they cannot reach the visceral organ. So what happen is here, they will form a synapse here, and the synapses are formed here in a ganglion. Where it is formed here? In the ganglion. So the neuron which is present before the ganglion, you call these as preganglionic neuron. Preganglionic neuron. So this is what the autonomic nervous system is made up of. This is what I'm telling you, that CNS will have a lot of nerves emerging from here, and these nerves will be containing neuron. So what it is made up of, these are three essential things CNS is made up of: CNS, ganglion, and where it is going, visceral organ. Okay. So here, though these organs are connected, what we want to do is we want to give information of CNS to the visceral organ. So for that we need some neurons or nerves to take the information. But since these neurons are not that long enough and the organ is so much away, for example, CNS wants to send information to your urinary bladder. So it's a long distance thing. So for that, first of all, the information will be sent to the ganglion, and from ganglion, another neuron will be taking information, and it will send information to the visceral organ. As a result, here a synapse is formed. So a neuron which is present before the ganglion, that's preganglionic, and the one which is present after the ganglion is a postganglionic neuron. Postganglionic neuron. Okay. Now, since autonomic nervous system, both in sympathetic and parasympathetic, have the same structure, but here the nerves emerging will be different, plus the neurotransmitter released on these synapses will be different. Okay. So if I'm talking about the parasympathetic nervous system, if I have parasympathetic nervous system, and parasympathetic and sympathetic, they are working opposite to each other, as I've told you in the previous lecture as well. They work opposite to each other. I'll give you one example. Visceral organ, if here is heart, and I tell you parasympathetic nervous system decreases the heart rate, and sympathetic nervous system increases the heart rate. Now how will they work? So from central nervous system, a nerve will go to this ganglion if we want to decrease the heart rate. A parasympathetic nervous system's neuron will take information from central nervous system, and it will be sent here. So now it has to give information to the another neuron. So how do the information from one neuron to another is transmitted? With the release of neurotransmitter. Okay. So here the neurotransmitter release will be acetylcholine. Now again the information is going to the visceral organ, and you know the information is always released from the neuron in the form of neurotransmitter. Again the neurotransmitter release here will be acetylcholine. So we say that in parasympathetic nervous system, the neurotransmitter will always released will be acetylcholine. What it will be? Acetylcholine. Okay. Now if I have sympathetic nervous system, and for example my body is in a position where I have flight and fight response. Okay. For example, I saw a snake, my body will react in a flight and fight response. There the sympathetic nervous system will switch on, and my heart rate will increase. How it will increase? From central nervous system, because the brain has judged the information, it's flight and fight, sympathetic nervous system will start, which will send information to its ganglion, and the neurotransmitter release here will be acetylcholine. Now this acetylcholine has triggered this neuron, and information is going to the heart, and here the neurotransmitter release will be adrenaline. As you know, adrenaline increases heart rate. It's also the hormone, and it is also a neurotransmitter. So at the postganglionic neuron near the visceral organ, here the neurotransmitter release in sympathetic will be adrenaline, whereas in the parasympathetic it will be acetylcholine. What's the full form of ACH? Acetylcholine. It's a neurotransmitter. Okay. So the question may be asked, tell me the neurotransmitters released in parasympathetic nervous system. At the ganglion, ACH. At the visceral organ, ACH. Tell me the neurotransmitters released in sympathetic. At the ganglion, ACH. At the visceral organ, adrenaline. Okay. So that's about your autonomic nervous system. Do remember, and these two nervous systems, sympathetic and parasympathetic, they work opposite to each other. They work opposite to. All right. Okay. Moving further to your sense organ. First of all, what is a sense organ? So sometimes when you are fighting with your friends, you are nonsense. You say you are nonsense, or what you say? Don't you have any sense? Now, what are these senses? What are these sense organs? So these sense organs, these can be a cell, they can be complete organ, they can be tissues that receive a sensory information. Okay, for example, you're looking at me. This is a kind of sensory information and it is received by your eye. So eye is a sense organ. You are hearing me. This voice is a sensory information and it is received by your ear. So ear is a sense organ. Your nose receives the impulse of a smell. Smell is a sensory information and the organ that is receiving is your sense. If I slap you on the face, though teachers are not allowed in these days. That's we have had a lot of slaps from teachers, even this ear pulling. But you guys are lucky. You have not faced, I think, all of these, isn't it? If I'm not wrong, or still in schools, teacher used to do this. I don't know. So, what happened is your if someone give you a slap, again, it's a sensory information, and it is received by sensory receptors in your skin. Okay, so these organs, they have certain things known as sensory receptor, and receptors are something that receives the stimulus. For example, your sense organ is nose. In the nose, we have sensory receptors, which are actually receiving the information. This nose, the skin of this nose is not receiving the information. Rather, there is an epithelium present inside the nose known as olfactory epithelium, which is sensing this information. So, sense organs, they contain sensory receptors. They contain sensory receptors that receive sensory input or information. Okay. Now these sensory receptors, they are of various kind. These sensory receptors are of various kind. For example, some are mechanoreceptor, depending upon what stimulus they are receiving. Some are mechanoreceptors. Some are thermal receptors. They will receive the information of temperature. Then we have chemoreceptor. Chemoreceptor receive the information in the form of chemicals. Okay, so what type of a stimulus they are getting, what type of a sensory information they're getting, we have classified them accordingly. And then we have the photoreceptor. You got to know photoreceptor will be having the stimulus that is light. Okay, so based upon the stimulus, based upon what? According to stimulus they receive, we have classified these sensory reception to various categories. First, we have mechano. Here, the stimulus will be mechanical stimulus like touch, like your slap. Okay. Thermal, we have temperature. In chemo, we have chemicals. And in photo, we have light. Photoreceptor are present in your eyes. Okay. In eyes, we have layer called as retina. That is a kind of photoreceptor. Whereas chemoreceptors are of two types. One is olfactoreceptor and another is gustato. Olfacto are present in your nose. They are for smell. They are for smell, whereas gustato are for taste. Okay. Then we have thermal, or the one that regulates or stimulates, or the stimulus is temperature. Like you touch anything hot, you got to know it's hot. You touch anything like ice, you get to know it's cold. Then we have the mechanical that receives the mechanical stimulus. Okay. So in the mechanoreceptors, we have different types of receptors. Like first is nociceptor. So nociceptor, also known as LGC receptor, is for pain. Yes. So, like I've told you, if you get slapped by someone, you feel pain. Or you get pricked by a needle, you feel pain. Why? Because on your skin, we have free nerve endings. And these are acting as receptors known as nociceptors. Okay, so these are for pain. Other example are your tango receptor. Tango receptor are for touch and pressure. Example for these is like Meissner's corpuscles. These are like cells, and Pacinian corpuscle. These are present in the skin. Where are they present? They are present in skin. Okay, this is also present in the skin, the free nerve endings. All right. So let's talk about the olfactory receptor, this olfactory receptors in detail. So these olfactory receptors, where are they present? They're present in the olfactory epithelium. This is the epithelium which is present in your nose. In your nose. So how do you smell? So let's see. Let's unfold this. So you have this epithelium present in your nose, and this epithelium contains different types of cell. What are these? First of all, very important cell, which is a sensory cell, olfactoreceptor cell. Olfactoreceptor cell, at one side it is innervated by this sensory neuron, and at another side it has this knob-like structure known as dendrite. Okay, which contains this small dendrite. This is dendrite, and this is knob-like structure. All right. So this is a knob-like structure, and these small hair-like structures are dendrite. Okay. Otherwise, it has also another cell like supporting cell. The function of supporting cell is to support the main cell, which is olfactoreceptor cell, which is olfactoreceptor cell. And then we have basal cell. So these olfactoreceptor cells, they are like bipolar neurons. What are they like? They are like bipolar neurons. We have done na bipolar neurons, yes. So they are like your bipolar neurons. And here we have this gland known as Bowman's gland. So there was a Bowman's capsule, and now there is a Bowman's gland. The function of this Bowman's gland is to secrete mucus. So that's why you have a lot of mucus. This is entire the layer of mucus in your nose. Okay. Now what happens is, for example, someone is cooking Maggie and you get a aroma of Maggie. So maybe in that food, or certain chemicals are present, they get diffused into the air, and from air you inhale them. So these small chemicals, they get dissolved in the mucus, and from mucus these chemicals, they bind to this dendrite. It bind to this dendrite. As it binds to the dendrite here, the depolarization of this neuron occurs. Like I have told you in the neurons, we need a stimulus to start the action potential. So here the stimulus is this chemical which attached to the dendrite. So I'm labeling it this chemical. When it gets attached to this dendrite, the depolarization occurs, and this nerve impulse will go through this nerve to your olfactory bulb. This is the olfactory bulb of olfactory lobe. And here the synapse is formed, and this information is taken through olfactory tract to your brain. To your brain. Which part of brain? To the temporal part of your brain, temporal lobe of your brain. Okay, so this is how you smell. So now you know how you smell. So some people who smoke a lot, their olfactoreceptor cells get damaged. That's why the sensation of smell in these people who are chain smokers is quite less. Okay, so that's about olfactoreceptor. Let's talk about your very important sense organ. Yes, that's eye. Let's talk about eye. So eye is a very important sense organ, and where it is present. Okay. And where it is present in socket of skull. In socket of orbit of skull. So in skull we have that small, round hall like structure like this here. So in this your eyes are present. How many eyes do you have? How many? Yes, one pair. So we have one pair of eye. We have one pair of eye. So definitely, what is the function? The function is vision. So your eyes, as I've told you, it's vital organ. So they are surrounded by fat tissue, and the function of this fat tissue is to provide protection. So it is surrounded by fat. For protection. So as you can see this structure, it's also the structure given in NCERT. So the eye is made up of three coats. There are three layers in the eye which it is made up of. Okay. So how many coats are there in the eye? Coverings or coats or tunics? Three coats. So I'm talking about three coverings or coat from outside to inside. So as you can see here, there is a this white color covering. This white color covering, this is known as sclera. The white portion that you can see, in fact, here also in your eye, that's your sclera. So the outermost covering or the coat of eye is sclera. It's a fibrous coat because it contains a lot of uh, fibers, collagen fibers. Okay, this sclera, if I say here it is white in color, it is known as white of the eye, and posteriorly it is very thick, and anterior it is very thin. It is so much thin it gets transparent, and this transparent structure name is known as cornea. What do you call it as? Cornea. So posteriorly sclera is thick and anteriorly sclera is thin, and anteriorly you call it as cornea. So this cornea is transparent portion where you put on contact lenses. It's present just in front of your lens. This is a portion that is quite changed during LASIK laser surgery to remove these spectacles, right? So this sclera is a white of the eye. White of the eye. Okay. Anteriorly, it forms cornea. It forms cornea. We'll be talking about cornea in detail. Just first we'll talk about the coats. Okay. Then we have the middle coat, which is blue-black in color. That is choroid. Choroid is vascular coat because it is, it has a lot of blood vessels. So the name is according to what they have. Fibrous because it has fibers, and vascular because it has a lot of blood vessels. And this has pigment called melanin. And due to this pigment melanin, it is bluish black in color. So this choroid, posteriorly it is thin, but anteriorly it is thick, just opposite to sclera. The sclera, it was anteriorly thin, but this one is anteriorly thick. It was thin and this is anteriorly thick. And it forms two structure. One is ciliary body and another is iris. So if you can see this diagram, I am highlighting with the blue color. This portion is choroid, it is thin here, but anteriorly it becomes thick, and this structure is known as ciliary body. And this ciliary body get extended further here and it forms the iris. So iris is that portion of your eye which gives color to your eye. So someone's eye are blue, someone's eye are little dark in color, so it appears black. ये काली काली आंखें, ये गोरे गोरे गाल, right? तीखी तीखी नजरें. Nazrein is different. We are not concerned here. Okay. So here, this iris gives color to your eye. Okay, iris gives color to the eye. And you know why there is a color? Because we have pigment known as melanin pigment. More the melanin pigment, darker the tone of the eye. If you have the least melanin, your eye will appear like greenish in color. If you have little more blue, little more brown, if you have more and the most one, then the eye will appear black in color. Okay. So there is iris present here, but in between the iris, this is the empty space you can see here. This space, empty space. What do you call it as? You call it as pupil. What do you call it as? Pupil. So this portion is pupil. P for pupil. So pupil is like a door of the eye. From here the light enters. Light enters here. From here. Light enters from here. So if I draw the eye and let me just draw it for you. If this is your eye. So in the eye, you will see this black portion here, the smaller one. And then if imagine someone has green eyes and this portion here like this. Green. Okay. So the black portion that you are looking out here is your pupil. This black portion is pupil, whereas this green one is iris. So iris have muscles. This controls the diameter of pupil. If these iris muscles, they contract and relax in a way that the pupil will dilate or contract. Dilate or contract, increasing the diameter. Okay. For example, if there is a lot of light coming out into your eyes, your pupil will contract. The pupil will get shortened. Why? Because more light can damage your eye. If there is less light, your pupil will dilate. The iris muscles will contract and relax in a manner that they will open and close this door. So dilation means you are opening the door, contraction means you are closing the door. So if there is more light, the door will be closed. If there is less light, the door will be open, so that more light will come and image will be formed properly. So this iris, it contains muscles. Contains muscles that regulate the diameter of pupil. Okay, so if there is more light, there will be, if there is more light, then there will be less diameter, or okay, diameter, or there is contraction, more contraction. If there is less light, then there will be dilation of. Okay, all right. So now we have the third layer, which is your retina. We'll be talking about this in detail. But before that, let's discuss a very important thing. And what is this? This is your ciliary body. So as I've told you, the choroid in the front, it gets thickened and it forms this structure known as ciliary body. So this is your ciliary body. Okay. This ciliary body, this is entire ciliary body. And here you have lens. Here you have lens. So ciliary body is attached to the lens, but it is not directly attached to the lens. It is attached to the lens with the help of these suspensory ligaments. What are these? Suspensory ligaments. Okay, these are suspensory ligament. So once there was a question that was asked, tell the arrangement of this. So is the lens directly attached to ciliary body? No, the lens is first attached to the suspensory ligaments and then it is attached to the ciliary body, and ciliary body is highly vascular. So as you can see, this portion is the lens. What about lens? Lens is biconvex in structure. Biconvex in structure. It's transparent and it is made up of crystalline protein. Which protein? Crystalline protein. The function of this lens is to take more light and helps in its refraction. Refraction. It helps in basically focusing your light, so that the light should come and it will focus directly on the retina. Let me show you how. So if this is your, boys and girls, this is your eyes white portion. This is cornea. This is cornea. Okay, follow me. This is lens. This is your choroid. And this portion is your retina. This is retina. Okay, this is retina. And this is your lens. And as I've told you, your lens is biconvex. What is the function? Focus the light. Transparent or opaque? It's transparent. What it is made up of? Protein. Which protein? Crystalline protein. Okay. Now, whenever, for example, here an object is present. First of all, the light rays will go to this cornea. It will pass through the cornea. This portion is cornea, which will also help in refraction. And from here it will go to the lens, and lens will try to form image here. And this portion where it forms the image, it's a yellow color portion and you call it as fovea. What you call this portion as? Fovea. This portion. Why it is forming image in fovea? Because it is a particular region where the resolution is maximum. That means clarity of image formation is maximum. And every time your eye wants to form image here only. Every time the eye wants to form image here only. Got it? Did you understood this point? Yes. So what this fovea basically is, it's a pit. It's a small pit. In this pit, a yellow colored area is present known as macula lutea. So technically, fovea is a pit. Fovea is a pit. In this fovea is present a yellow pigmented area known as macula lutea. So fovea is a pit, okay, and this is the area known as macula lutea. It's yellow in color, okay. So why here the image is formed, this will I'll tell you later on. Now there is a portion here where you can see there is no retina. Retina is this pink layer. At this portion there is no retina, and this point is known as blind spot. What you call this point? You call this point as blind spot. In this blind spot, no image is formed. No image is formed. Why? Because here there is no retina, and when we have no retina, no image will be formed here. But from the blind spot, but from the blind spot, the optic nerve will leave the eye and blood vessel will enter. From this portion, particular portion, the optic nerve is leaving, because definitely it's a sense organ, it will be innervated by the nerves. Here the optic nerve will leave and blood vessels will enter like this. So this is blood vessel and this is optic, okay. So there is a line written in NCRT about the position of fovea. So it is said that fovea is present at the posterior pole. This is the anterior side of the eye, this is the posterior side of the eye. Yes, it's present at posterior side, lateral to blind spot. So if this is a blind spot, it is present laterally to it. That means on the side of a blind spot. Okay. So it is said that what's the exact location of fovea? On the posterior side of the eye, where lateral to the blind. Okay. Now let's talk about this portion called cornea. The cornea, it's basically a part of sclera only. It's thin, it's transparent and refractive. It helps in refraction of light. Very important point to note that it is avascular. That means it will be having no blood supply. Vasa word is used for blood vessel. There will be no blood supply. Hence, the transplant of cornea is quite easy because it has no blood vessel. There will be no blood. There will be no antibodies, no WBCs, and no graft rejection. And this is rich in collagen fibers. Which fibers? Collagen fibers. Alright, do not forget about the cornea. Apart from cornea, your eye is also covered by another very protective layer known as conjunctiva. So there is also another layer known as conjunctiva, and it is vascular. This one is vascular. So whenever you get eye flu, usually your conjunctiva gets inflamed, and we call this disease as conjunctivitis. What you call it as? Conjunctivitis. Now let's talk about very important point, that is retina. Let's talk about retina. So as you can see in this diagram, there is lens. So before lens, there is a chamber formed here, and after lens, there is a chamber here. So this chamber is known as aqueous chamber. It contains a fluid known as aqueous humor. Contains aqueous humor. Okay. Aqueous humor is water-like fluid. It is water-like. And this is vitreous chamber. And vitreous chamber contains gel-like fluid. It contains gel-like fluid. So the function of this fluid is it helps in refraction. Helps in. Alright. Okay, let's move further. So if I talk about retina, I told or I have named sclera as fibrous cord because it has a lot of fibers. I called choroid as the vascular cord because it has a lot of blood vessels. Okay. Now let's talk about the retina here. So if I talk about retina, retina is known as neural coat because it is basically related with neurons. So what's the structure of retina? First of all, if this is your choroid, and from here the light enters, first of all, you will be having here pigmented epithelium. What will be here present? Pigmented epithelium. Pigmented epithelium has a function that it will prevent the reflection of light, just like your camera have a black screen behind. Okay, so here pigmented epithelium. So the first layer is pigmented epithelium. This is your choroid. And from here light is entering. Pigmented epithelium. Below the pigmented epithelium are present photoreceptor cells. So I'll be drawing just one layer. Okay. So photoreceptor cells are of two types. One looks like rod. So their name is rod cells. And one look like cone. So their name is cone cells. Okay, let's change the color. So these are cone cells because they look like cone. And other, they look like rod. So their name is rod cell. So these are both photoreceptor cells. What is a photoreceptor? That means these are sensory receptors of the eye. Okay. So these both are rods and cones. So these are rods, these are cones. They are collectively known as photoreceptor cells. And they are forming because they are a kind of bipolar cells modified. So they are forming synapse with the other neurons here. And the neuron here present are bipolar neurons. These are bipolar neuron. And these bipolar neurons are further forming the synapse with the ganglionic neuron. And these ganglionic neurons are attached with the optic nerve. They are attached with what? Optic nerve. So these are bipolar neuron. And this is ganglionics. Okay. So most of you gets confused with the arrangement of these cells. So guys, do remember the arrangement is just like this. From here the light will enter. Light will pass on to these cells and then it will stimulate these photoreceptor cell. So some of you think, ma'am, light has to work on photoreceptor. Why not photoreceptors are here? Or you are teaching wrong. Yes, there are kids, they say like these things. I feel really bad. So anyways, so light always go from this position. Okay. And here at the end photoreceptor cells are present. Now what does these photoreceptor cells have? They have photopigment. They have photopigment. What is this photopigment? Let's see. So photopigment in rods and cones is different. Okay. So first I'll talk about rods. So rods are the type of photoreceptor cells which are responsible for twilight vision or moonlight vision. Okay. Basically, it helps us to see in dark. In dark, you cannot see. Okay. So if there is a room with no light, you cannot see. So I cannot say rods helps in that dark vision. I will say it will help you to see in dim light. In which light? Dim light. So it is helping in dim light vision or also known as scotopic vision. Their number is more. They are more than cones. They are more than cones. Okay. So what pigment do they have? If I'm talking about rod, what pigment do they have? They have a pigment known as rhodopsin. What is the name of pigment? Rhodopsin. So this pigment is made up of two things. What is retinal? Retinal is formed from vitamin A. It's a derivative of vitamin A. Vitamin A is another name is retinol. It's alcohol. But when it gets converted into an aldehyde, it becomes retinal. So retinal is an important part of the photopigments, whether it's in cones, whether it is in rods, and it's a derivative of vitamin A. Now, another thing here is a protein known as opsin protein. So in case of your rods, this protein is known as scotopsin. This protein name is known as scotopsin. All right. And what's the color of this pigment? It is purple in color. Purple in color. Okay. All right. Moving further. If I talk about cones, cones help in bright light vision. Bright light vision, or we also call it as photopic vision. It helps us to see colors. It helps us to see colors. So now you are looking at, this is black color, this is white, this is orange, this is black. Okay. So this is skin color. So all these colors you can see with the help of cones. Okay. So these cones, they have a pigment. They have a pigment. And what is the name of its pigment? Idopsin. So like rods had rhodopsin, it has idopsin. It is also made up of retinal, which is an aldehyde derivative of vitamin A, and protein known as opsin. But here, since you want to see colors and you want to see in bright light, so options are of different kind. Options are of three kinds. One is erythropsin. Erythropsin is for red color. Chloropsin. Chloro word is used for, yes, in botany, and botany we always use green color. Plants color is green. Okay. So this is a pigment for green color. And then we have cyanopsin, which is a pigment for blue color. Now you will say, ma'am, there are only three pigments, but I can see black, I can see white, I can see purple, I can see all the colors. So guys, what type of pigment is stimulated by the type of wavelength that color is produced? For example, in lower classes, when you started doing paint, you got to learn about primary colors, right? Then you mix the colors and you form different shades or hues. Like if I match the green and red, it will become brown. So if the light is stimulating both these pigment, you can see brown color. But if the light is stimulating only red pigment, you can see only red color. Okay. Now what happen when the light strikes these photopigments? So we'll see, if I have rhodopsin here, in rhodopsin, retinal and opsin are joined to each other. They are like lovebirds. You cannot put them away. They are like, they will sit together only. Okay. Now when the light comes, these two has to dissociate. Okay. So they get dissociate. Why? Because there is change in conformation. For example, if two lovebirds are sitting like this, holding hands, but suddenly the teacher comes in the class, they will leave the hand and they will separate. They will fall apart, right? Right. Why? They are changing their position. Just like that, opsin changes its position. Sorry, retinal changes its position, and hence they have to move apart. So when they move apart, there is a stimulus that arises in photoreceptor. A kind of stimulus is received to the photoreceptor, and hence it will generate action potential. And when it will generate the action potential, this potential will be sent to bipolar neurons through synapse, then to ganglionic, and then to optic nerve, and then to brain. And the image is formed here. Okay, so this is how a light can stimulate the dissociation of these retinal and opsin. But now you will say, ma'am, if these pigments are broken down, so how we maintain the vision for all these years? Your eye must have destroyed. Why it is not destroyed? Because after the action potential is generated, this goes reverse, and there is an enzyme called retinal isomerase, so it will associate them together. Like the teacher goes away after the class has finished, the lovebirds will again join, just like this. Okay. All right. So now, not every time this action potential is generated. I means to say, sometimes depolarization occur and action potential will be generated. This happens during dark. So action potential is generated when there is depolarization, right? And we are talking about rhodopsin. That means if I'm saying action potential is generated, must depolarization has taken place. And depolarization takes place during dark. What if I am explaining you about cons? If I'm explaining about cons, I will never use the word depolarization here. There, with the association of retinal and opsin, the hyperpolarization occur. So whenever the hyperpolarization occur, you are talking about the bright light. So during bright lights, the cone undergoes hyperpolarization, and during dark, your rods are undergoing depolarization. So depolarization will cause the it gives the stimulus of dark. Okay. All right. So this is about the eye. Now let's talk about fovea. As I've told you, why the image is formed here and why it is of maximum resolution. Because here in fovea we have only cones present, and cones help us to see in bright light. Yes, cones help us to see in bright light, so that's why the resolution is maximum here. So for example, you just perform this experiment at home. Okay, if you put a pen here, so here you have fovea just behind your lens, and the image formed is exactly clear. Just focus on pen. If you focus on pen, the outside things, they are quite blur, because their image is formed on these sections, whereas pen's image is formed here. Okay, now move the pen. Move the pen. Now, whatever is present in front of you, its image is formed on the fovea. So now if you want to form the image of a pen to the fovea, you will move your head. And now the pen is again in front of fovea. So this is how you move your head. And if you want to focus it, just like camera gives a focus, like there is a portrait mode in the camera. If you don't know, no problem. Just like that, whatever you are clearing, you are focusing on, it's quite clear image is formed. Its image is formed here. Others are blurred because their image is formed on the other areas. So it's not like that other areas are not forming an image, they are also forming the image. Okay, and the image formed is always, yes here, inverted. Then the brain will process it in and erect it. Okay, all right, now let's talk about ear. Okay, another sense organ. So let's talk about ear. So ear is the another sense organ that help us to hear and balance. So the two function that your ear perform is hearing and balance. So if I talk about various parts of your ear, there are three parts in the ear: the outer ear, the middle ear, and the inner ear. Okay, three parts. So the outer ear consists of ear pinna, external auditory canal. Just I'm telling you about the names of all these. How they look like, we'll be doing it, okay? And meatus as well, the opening of the canal, and the tympanic membrane. And the tympanic. Okay, so we have three parts in the outer ear. In the middle ear, we have tympanic cavity, and in this tympanic cavity is a space. We have three ear bones known as ear ossicles. So we have done these bones in the locomotion and movement. Remember, what are these? Malleus, incus, and stapes. Then we have the inner ear. This inner ear is also known as labyrinth. And labyrinth have two portion, outer and inner. Outer portion and inner ear. Otherwise, if we see structurally, this is a basic structure of inner ear that we say it's look like a labyrinth. Labyrinth, you understand, in Hindi we say bhool bhulaiya, right? So there is an outer part of labyrinth and the inner part. But actual parts in this are the vestibule and cochlea. So vestibule and cochlea are in the form of labyrinth, in the form of bhool bhulaiya. Okay, let's see how does it looks like. Okay, you must have seen this diagram in NCRT, if I'm not wrong, right? So here you have this ear pinna. So in the outer ear, we'll see the outer ear. We have pinna. Then we have this canal. This canal is filled with hair and gland. What gland? The wax gland. So here in the auditory canal, you will see some glands. And what is this gland? Wax gland. And this opening is the meatus. This is the external auditory meatus. And this will then this thing has this particular membrane at the end of the external auditory canal. There is a membrane known as tympanic membrane. What membrane? Tympanic membrane. So this tympanic membrane is also known as eardrum. So up to this is your outer ear. So from here to here is your outer ear. Then we have middle ear. So middle ear is present in this cavity known as tympanic cavity. This tympanic cavity is attached with this tube known as the Eustachian tube. The function of Eustachian tube is to equalize the pressure. Equalize the pressure. Now what pressure? So here you have this tympanic membrane. Let me just color it. So this tympanic membrane is like a door, the closed door which never opens. Okay. Here outer air is coming and it has different pressure, but the pressure air of the air in the tympanic cavity will be different. So we want the pressure of air which is coming out here and here should be equal. So what we did, we put a tube here, and this tube opens into pharynx. And in pharynx, we engulf air. Like I'm talking, I'm engulfing some air in my mouth. And in mouth, later, in the after buccal cavity, we have pharynx. And there in the pharynx, we have a eustachian tube which opens into the middle ear, this cavity of middle ear. And here we have three bones, the malleus, incus, and stapes. The stapes is the smallest bone and it opens into a window here. As you can see, there is a window. This is known as oval window. And we call this oval window as the door to inner ear because it open into the inner ear. Door to inner ear. So if someone asks you what is a door to inner ear, what will you say? That's your oval window because it is attached to your these bones. That's a middle ear. Now as you can see this structure, the cochlea. You can see this cochlea and this portion which is vestibule. These are the part of your what? These are the part of your inner ear. Okay, so I'm just right now I'm just showing you how the structure looks like, what they do, and their detailing. We'll be doing it further. Okay, now as I've told you, the inner ear is made up of vestibule. This is vestibule, and cochlea. This is cochlea. Okay, and outside this is present the bone which is a temporal bone. So your ear is basically embedded in your temporal bone. Which bone? Temporal bone. So if I consider this entire as a labyrinth, so labyrinth is consist of two things, the outer labyrinth and the inner labyrinth. The outer labyrinth is formed of a bone. So this entire structure, outside structure, is made up of a bone. So we say the outer labyrinth is made up of bone. It's bony. And inside we have membranes. Inside what do we have? Membrane. So the inner labyrinth is made up of membranes. What it is made up of? Membrane. Okay. And here we will be finding certain, you'll see certain fluids are present here. This fluid is perilymph. And the fluid present here is endolymph. Okay. I think you all understood this. Basically, you consider this entire two structure, the vestibule and cochlea, as labyrinth. And labyrinth thing is divided into two parts. This is the outer labyrinth, which is made up of bone. And then we have membranous labyrinth, which is made up of membranes. What membranes we'll be talking about later. Now, let's see, how do you hear? How do you hear? If this is a ear pinna, the sound waves will come here through the ear pinna and it will strike the tympanic membrane. And now the tympanic membrane will vibrate, ton, like this. Now, the voice is very low. You want to amplify it so that the information should go nicely inside. Information should go nicely. Okay. So, as the information has gone here towards the tympanic membrane, now the malleus, incus, and stape, they will start to dance. They will start to dance. There is a leverage system here, and they will start to move and they will amplify the sound. So amplification of sound takes place by ear ossicle as well as by the oval window. Oval window functions just like that. When you if you play a song on the phone and you put hand on your speaker, you are narrowing the speaker, right? As a result, there will be more sound. Just like this, the oval window is functioning and it is also amplifying the sound. So these bones and your oval window both are amplifying the sound. Now the sound is going to enter into the inner ear. Now what will happen inside the inner ear? So for that you need to understand the structure of inner ear in more detail. So let's draw the structure of the inner ear. Oh ho, how can thank you appear so fast, huh? So if I have this structure, I have opened the cochlea. Vestibule's function is in the balancing, but cochlea's function is in the hearing. So the outer labyrinth is made up of bone. Yes, this is bony structure, and inner labyrinth is made up of membrane. You must have seen that toy that you have bought that in your fairs. When we were little, our parents used to took us to fairs for all those swings and all, and there was a toy. If you there was a whistle and a coil portion with that. So when you blow the air and you start blowing the whistle, that air will go into that particular coiled portion and that coiled portion get unwind. Have you ever seen that? Have you ever played with that toy? Tell me, tell me. Yes. So it's just like that. So cochlea is just wind portion. I have unwind it. So when you unwind it, you will see it appears like this. So externally, you will see this portion. This is a bony portion, outer portion. This is made up of bone. Okay, and here we have this portion which are membranes. Okay, now this what is the name of these membrane? This membrane's name is Riesenars membrane, and the name of the membrane below is Basilars membrane. What is its name? Basilars membrane. Okay, so due to this membranes formation, there is a demarcation in the chamber. So this chamber will be known as scala vestibuli. This will be known as scala media. Scala media is a middle portion and it is known as cochlear's duct. And the lower portion is scala tympani. Okay. So likewise, I've told you the outer labyrinth and there is a membranous labyrinth. So here the fluid present will be perilymph and here the fluid present will be endolymph, just like that. So if you see the NCRT, you will come across a structure which is formed in this manner. They have made it like this. So this is the Riesenars membrane. This is Basilars membrane. Here you will find perilymph, and here you will find end. Okay. So what we have done, they have taken this section. We are taking this section. That's it. Okay. So in the Basilars membrane, an organ is present which is known as organ of Corti. Have organ of Corti. Okay. So let's see how the organ of Corti it looks like. What does it have that helps you to hear? So if I draw the diagram of organ of Corti, what does it have? In the basilars membrane, we have certain cells present here. And above the basilars membrane is present a membrane called tectorial membrane. So if this is a basilar membrane, it will be having cells which are known as hair cells. What are these cells? These are hair cells. Along with hair cells are present certain supporting cells. These are supporting cells. So the cells which are present towards inside, the inner hair cells, and the cells which are present towards outside, they're outer hair cells. And this membrane is tectorial membrane. Tectorial membrane. This tectorial membrane is made up of glycoproteins. Glycoproteins. Okay, now what happens is your sound waves, they will enter into your ear, then external auditory canal. Okay, let's just write it. First, it will enter into the ear pin, not into the ear pinna, external auditory canal. Ear pinna will just collect the sound waves. External auditory canal. Okay. Then these sound waves will hit the tympanic membrane and these sound waves will be converted into vibrations. The vibrations are formed. Now these vibrations, it will enter into middle ear and hence its amplification will take place. So the amplification of sound waves will take place here. The amplification will be taken place. It will be by the ear ossicles. And by what? By your, yes, the oval window. After the amplification has taken place, these vibration, they will enter into, for example, if we have drawn this diagram, imagine the oval window is present just right here. So from oval window, the vibration will enter here. Yes, from oval window. Okay, let's do this diagram first. So vibrations, so the sound waves have entered here in tympanic membrane. They will hit and they will be converted into vibration. Now here it will be amplified and this is oval window and oval window opens into your scala vestibuli. Okay, here oval window opens into scala vestibuli. Now the sound waves have entered here. From here the sound waves will move downwards. It will also move from here to here and also from here to here. Ultimately, all the sound waves have a similar function that they will have to enter into scala media. That's the cochlear duct, which contains the organ of Corti. So organ of Corti is just present right here. If this is a scala media, so organ of Corti will be present here. Okay. Now what will happen when the sound waves enter? So it will lead to the movement of fluid. What fluid? The perilymph and endolymph. Here, the fluid present is your endolymph. The fluid present is your endolymph. Yes, here what is present? Endolymph is present. So when the vibrations come, it will lead to the movement of this endolymph. And when the endolymph it moves, these hair on hair cells will move. These hair on hair cells will move. Okay. These hairs are basically stereocilia. These are like stereocilia. Cilia, you know, hair like projections on cell. How it is different from stereocilia? Cilia can move because it has motor apparatus here. Stereocilia cannot move on its own. It will move when someone will give it force. So here the force is that vibrations which are moving endolymph. Now amplification has taken place. Now what will happen? Vibration enters inner ear. Now what will happen? Movement of lymph. Which lymph? Endolymph. And when this will move, there will be movement of hair. Okay, hair is known, or it is another name for stereocilia, which will lead to depolarization. Because ultimately you need a stimulus. So the stimulus is the hair will move and it will touch the tectorial membrane. So when the hair move, it will touch the tectorial membrane. This will act as the stimulus here. So what's the stimulus here? The stimulus is given by movement of hair, which will be rubbed by tectorial membrane. Okay, just like this, if this is my hair, and imagine this is a hair of your hair cell. Imagine I blow hair on the hair, like this. Okay, so now I have tectorial membrane here. Now I will blow it. So it is rubbing on this tectorial membrane, right? So just like that, this hair cells, because they are the receptors here, they are attached to the nerves here, like this. So when it will rub, the action potential will be generated, and through these nerves, they are going to which nerve? Auditory nerve. They will send the impulse to the brain. Okay, I hope this is pretty clear, and this is how you hear, and this is how you hearing my sound. Now let's see the another function which is done by your uh, vestibule, that is balance. So the function of vestibule is to balance, equilibrium and balance. How does it happen? So let's draw the vestibule for it. So if you see the vestibule have these structures, three structure. You call them as semicircular canals. What you call them as? Semicircular canals. So these are semicircular canals. Okay. These semicircular canals, at the base, they have these swelling portions, and these are known as ampullae. What you call them as? Ampullae. Okay. So these which looks like ear, like of a Mickey Mouse. So these are what? These are semicircular canals. Semi means because they are half circle. At the base of every semicircular canal is present a swelling like structure known as ampulla. This ampulla contains cristae. It contains cristae. Cristae contains sensory receptor which helps in balancing. And below this is present this structure, and here the cochlea starts. Here we have two structure. One is utricle, another is saccule. So here you have two structure. One is utricle and another is saccule. This utricle and saccule, they contain another structure, just like ampulla contain cristae. They contain otolith organ. So utricle and saccule contain otolith organ. Okay, they contain what? They contain otolith organ, and these otolith organ is present in structures known as maculae. In maculae. Okay, like in ampulla we have structure called cristae which contains sensory receptor. Just like that, the maculae are present which contains the sensory receptor. Let's see how does they work. If i say if i say we have here structure called as ampulla, and then we have structure called saccule. Ampulla helps in your rotational acceleration and deacceleration. That means if I'm moving like this, my ampulla are helping in balance. But if I'm moving like this or I'm standing here, then who is functioning here? The otolith organ or macula are working. Okay, how does they work? Let's draw the ampulla first. So ampulla, obviously, because it has sensory receptor, it will be having hair cells here. It will be having hair cell. So these are hair cells. You will find hair cell in all the sensory receptors of your ear. In ear, the sensory receptors are hair cell. If the question is asked, what are the sensory receptors present in ear? You will say they are hair cells. So these have here supporting cells with them. Okay, like this. And above them is present a jelly like fluid known as cupula. What is this cupula? Gel like. So you must have heard while in the in lower classes, or while you're talking to friends, when we rotate there is a fluid in our brain that rotates, that's why we feel like this. So that fluid is your cupula. So when you move like this in this direction, this cupula moves, and when this cupula moves, the hair cell will be stimulated. This is hair, this is hair cell. And when the hair cells are stimulated, they will get depolarized, action potential will be generated, and the information will be sent to auditory nerve. And auditory nerve will send to which part of the brain? Yes, equilibrium and balance is done by cerebellum. Now, second thing that you have is maculae. Maculae. How maculae looks like? So maculae also have hair cells. As I've told you, hair cells are important in your ear. Again, they are also supported by supporting cells, but the supporting cells are little curvy. Okay, so these are supporting cell, these are hair cell. Okay, now here is a present a membrane, and on membrane are present calcium carbonate structure known as otolith organ or otoconia. Okay, so this is a membrane, and above membrane is present the structure known as otolith organ or otoconia. These otolith organ or otoconia, they are made up of calcium carbonate. Okay, so for example, you're sitting in a car and suddenly a brake is applied. You move like this, but then you get back to your position, right? What happened at that point is, when a car brake is applied, your stationary position is this, but the car has stopped and you move like this. At this point, your otoconia will also move. They are free. Hair cells are fixed. These otoconia, they are free. When the brake is applied, your hair cells will be present just here, but otoconia will move like this. So when the otoconia moves like this, they get touched by these hair cell, and hair cells get stimulated, and the information is sent through auditory nerve to which part of brain? You all know, cerebellum. So cerebellum will then get this information, and cerebellum will now send information to your muscles and ask the muscle to get back to the origin. This is how the balancing is done. Yes? Okay. So these are just sensory receptors. How they're helping in balancing? They're helping in sending information to the brain. Okay? Or how are they helping? They're sending information to the brain. The rest thing of balancing will be done by your locomotion movement, which will be done by muscles when the muscles are given signals by the cell. So this is how you get balance. All right, so let's do PYQs, boys and girls. Let's get started. Good vision depends upon the adequate intake of carotene-rich food. Select the best option from the following statement. Very smart question. Vitamin A derivatives are formed from carotene. The photopigments are embedded in the membrane disc of the inner segment. Retinal is a derivative of vitamin A. Retinal is a light absorbing part of all the visual photopigment. A option says A and B, B says ACD, C says AC, and D say ABCD. Do it. Yes, yes, yes. So in this question, as you can see, the first option says vitamin A is derivative and are formed from carotene. Yes, carotene is present in carrot. We always say eat carrot and eyes will be good, and we have already done it in the previous chapters that this carotene gets converted into vitamin A. Okay, in the biotechnology. So vitamin A is a derivative. True. Okay. True. Photopigments are embedded in the membrane disc of inner segment. This is something new for you. Retinal is derivative of vitamin A. True. Retinal is light absorbing part of all the visual pigment. Very true. It's present in all the pigments. Now what happen is, if this is your rod, okay. So now rod is divided into two segments. This is the outer segment and this is the inner segment. So these are always embedded in the membrane of which segment? Of your outer segment, not the inner. So these pigments, which where are they actually present? They are present in the outer segment. So A, C, and D are correct. So answer is B. Next, in transparent, the transparent lens in the human eye is held in its place by very simple. If this is your lens, how it will be present in place? By smooth muscle attached to iris? No. Ligaments attached to iris? No. Ligaments attached to ciliary body? True. As I've told you, the lens location is how it is located there. These are ciliary body and it is attached to the lens with the help of these ligaments known as suspensory ligament. So answer is C. Next. Photosensitive compound in human eye is made up of? Simple. Opsin and retinal. The photosensitive pigment or photopigment is formed of your one protein opsin. These two are lovers, I told you. So answer is C. Next. Choose a correct statement. Nociceptors respond to change in pressure. No, nociceptors are for pain. Measles corpuscles are thermoreceptor. No, I've told you they are tango receptors for pressure and touch. Photoreceptors in the human eye are depolarized during dark and become hyperpolarized in the light stimulus. Receptors do not respond graded potential. Yes, they do respond to graded potential. What is a graded potential? That is change in the potential difference. Because there is change in potential difference, that is why they're getting depolarized. They are getting action potential. So yes, I've told you during darkness, your eye gets depolarized and during light, it gets hyperpolarized. So answer is C. Next. Destruction of the anterior horn cells of the spinal cord will result in loss of. Very simple question. If you are confident enough that you have done the structure of spinal cord, this question is simple. You don't have to get confused by the option because the options are confusing here. Let's see. Integrating impulses, secondary impulses, voluntary motor impulses, commissural impulses. See, we have drawn this spinal cord. In spinal cord, there was DPS, Delhi Public School, dorsal side, posterior, which is sensory. Then ventral, anterior, which was motor. So it is saying if we destroy this portion, tell me which impulses will not go. You know, it control motor, so you don't have to think of other things. The examiner, whenever it put or make a question, right, it can put anything in the wrong options. So you don't have to bother about if you know the wrong option or not. Have you heard of these or not? You are not concerned with that. You cannot learn each and everything of biology present in this entire earth. Okay. So if you know it, if this is destroyed, motor impulse will weak, you don't have to do anything of it. Do not get disturbed or get panic in exam. This can occur in the exam. You may come across about these things, type of things. Okay, so answer is C here. Be confident enough and put C. Next. A gymnast is able to balance its body upside down even in the total darkness because of what organ helps in balancing? This helps in hearing. Cochlea also helps in hearing. Tectorial membrane is also responsible for hearing, whereas vestibule helps in balancing. What's happening? Everything is C. Every answer is C. In mammalian eye, the fovea is the center of the visual field where more rods are than cones. No, there were only cones present in fovea. That's why in bright light the resolution is maximum. High density of cones occur but no rods. Very true. Optic nerve leaves At blind spot. Only roads are present. No. Answer is B. So the monotony has been broken. Now from C we have turned to B. So I think that's a repetitive question. So leave it. Next. So this is a diagram they have given. So make sure you know the diagrammatic questions. First, B is blind spot. B is blind spot has a very few rods and cones. No, here rods and cones are absolutely absent. C is aqueous chamber, reflects the light which does not pass through the lens. D, choroid. Where is D? This is no, this is sclera. So this is incorrect. Okay. And A is retina, contain photoreceptor rods and cone. Very true. C is aqueous chamber, true, reflects the light which does not pass through the lens. It does not help in reflection, it helps in refraction. So the answer is D. Next. Which one of the following statement is not correct? Retinal is a light absorbing portion of visual photopigments. True. Retinal is a pigment of, or it's a light absorbing portion. In retina, the rods have the photopigment rhodopsin, while cones have three different photopigments. True. Rhodopsin have only one type of pigment or one type of protein called opsin, whereas cones have chloropsin, cyanopsin, erythropsin. Retinal is derivative of vitamin A. So this is incorrect. So answer is C. Again, C has come, yaar. Rhodopsin is a purplish red protein present in the rods only. Very true. What's the color of the this protein? Purple color. So answer is C. Okay, I have written already. Next. Which part of the human ear plays no role in hearing as such but is otherwise very much required? That is vestibular apparatus. True. It has nothing to do with hearing. Ear ossicles amplify the sound waves. Eustachian tubes equalizing pressure. Organ of Corti hearing. So A is vestibular. Next. So this was it about PYQs. It was a great journey with you guys. We have worked so much hard for you. So much hard. We knew that there are a lot of students out there in India who really do not understand Hindi and they don't find comfortable in the teaching with the Hindi or English type of English and Hindi. So that's why we have given you this series so that you can do good in your NEET examination. In fact, those who are giving their 12th, they might have find it productive as well. So this was all for all of you who cannot afford it and they really want to get good scores in NEET. So if you really put your 100% and rely on these videos, trust me, you can get good scores in NEET. The only thing that I would like to tell you how to prepare for NEET. So here let's since you are my students and I have got so much attachment with you guys now, and a lot of people who are from various areas, I always see comments. You're giving so good comments, and sometimes I feel overwhelmed. But still, again, we have not reached so much students who are in help and they don't understand Hindi. Probably, I don't know, they don't know about us. They don't know that yes, there is someone who is with them and want them to get good scores. But still, anyways, how you will prepare for NEET? First of all, whatsoever lectures you are referring to, for example, I am thinking you are referring my lectures. So first of all, go for lecture notes and lecture. Okay. Then read NCRT and solve PYQs. That's it. Three things you have to do, but give your 100% in these. Give your 100%. Now, what you have to do is, if you are making notes, highlight everything that belongs to NCRT. If from so long you are referring NCRT, you can also make NCRT as a basic notes and add things which I am telling you. So you should have only one notebook or a book which will contain everything so that you can revise it at last. Okay? Yes. Another thing, prioritize your chapters. Chapters priority should be from highest question asked to lowest question asked. Now, segregate them. Segregate them. Which one is toughest for you? From toughest to easiest. What we do is, for we whatever thing we know, we first of all pick those things up. Like if I know my chemical control is so good, I will start doing it with chemical control. But what I'm doing is I'm wasting my time. I know already chemical control, I know all the hormones. Why am I wasting my time? I should pick the chapter which is toughest for me. For example, I feel neural control is the toughest of all. I will give my 100% to neural first, then I will go with the second toughest, third toughest, and then I will go with the easiest one. So, some people or some students, what they do is they pick the easiest first because they find it good. They find it, pleasurable. But that pleasure is for moment. You want to target NEET. So, for that, pick your toughest chapter and then go for the easiest one. And must go for PYQ. At this point of time, since you have not much time, do not go for any other reference books. They will confuse you up, right? Just go for PY. Okay, so this was it. It was a great journey again. I'm telling you, thank you so much. Thank you so much for showering a lot of love. And this is your Diksha ma'am. And a lot of students were asking, ma'am, what is your full name? My name is Diksha Sharma. And this was all it. This is my army, Diksha ma'am's army. And I just wish you all the best. And I will keep showering a lot of, videos, conceptual videos in the form of shorts or short videos so that you can revise the things again and again. Just keep following what we are teaching you. Believe in yourself. Believe in yourself. The only thing that you can do right now is to believe in yourself. Just these months will decide what you are going to be. A lot of you will start panicking. Only few months are left. Few days are left. We cannot do it. We have not done anything right now. We have not taken any coaching. Trust me, if you have not done anything, even if you do not have undergone any coaching, even still, if you work hard, give your 100%, be true to yourself, and cover each and everything, you can clear NEET. I can bet it. You can clear, right? So trust yourself. Trust that you can also do it. You're not less than others. What if you're not that privileged? What if you're not that privileged? Doesn't matter. Your will decide what you're going to be. So I've seen students who are highly privileged. They have a lot of things, lot of gadgets, every notebook, every book, great tuitions. Still, they cannot make it in the NEET. The thing is, they do not have a will to do that. They do not have a will to do that. The one thing that keeps you high and want that thing that will lead you to your goal is your will, whether you want to do it or not. So here I conclude this series, but you, as I've already told you, that I will bring lot other videos for you. For that, stay tuned to our channel and keep working hard. Keep working hard. This was Diksha ma'am, your zoology teacher. Take care. Bye bye. All the best. So much love.